Hello and welcome back to Intro to Biology for Majors, or BIOL 1224. This is the Chapter 4 PowerPoint from your Openstax book, Biology 2E. This PowerPoint is Chapter 4, Cell Structure, and this is the final chapter of the first module of this course. So as always, we're going to start off talking about our objectives. This chapter has 3 objectives, and remember the chapter is about cell structure. So the first objective is to recognize 2 types of microscopes for observing cell structure. The second is to analyze the differences in prokaryote and eukaryote structures. And the third is to identify organelle structures within cells. So in this slide, we're talking about how cells are the units of life. So a cell is the smallest unit of life that can function independently. Most cells are too small to be seen by human sight, which is why we use microscopes. So a scanning electron microscope is a very powerful microscope that reveals extremely small cellular details. And if you look at the right side of your slide, I put up a couple of pictures. Just this one on top is called a scanning electron microscope, or SEM. And normally this microscope on the left is the actual machine. They're almost always connected to a desktop or computer because the images are so detailed and so small that you have to be able to view them on desktop rather than just through an ocular, like on a regular light microscope. So that being said, light microscopes are less powerful than electron microscopes. They can generate images of living cells. And in the labs that you all have with me, we use light microscopes that look a lot like the one on the slide. So some of my notes for this slide are that atoms bond together to make molecules, and macromolecules form organelles. These organelles work together to maintain an environment known as a cell. Cells are considered the building blocks of life, or the fundamental units of life. The smallest living things are single-cell bacteria. Teaching microscopes are generally light microscopes, which is what I was saying here, that teaching and using them in lab, we're going to be using light microscopes that look a lot like this. Scanning electron microscopes move a beam of electrons back and forth across the surface of a sample to create a detailed 3D image. And people who do this professionally, they're working in something called microscopy. And microscopy is just, working with microscopes in order to look at extremely small units within the cell. There are some people that do this at OSU Center for Health Sciences and medical research. It's a really common thing for people to do. So let's start off by talking about prokaryotes, but I do want to do just a short review of some of the slides that we hit on in previous slideshows because they are relevant here. So prokaryotes are predominantly single-celled organisms of the domains bacteria and archaea. And in some previous slideshows that we viewed, we did talk about 3 different domains that we're really digging into in this course, and that is bacteria, archaea, and eukarya. So bacteria and archaea, as mentioned previously, they used to be one shared domain, but bacteria are actually more commonly found in day-to-day life, while archaea are found in extremely harsh environments, so they were separated for that reason. So prokaryotes are single-celled organisms, which in turn means that they're simple organisms. All prokaryotes have plasma membranes, cytoplasm, ribosomes, and DNA that is not membrane bound. So in these prokaryotes, DNA is actually free-floating rather than being bound in a specific spot within that prokaryote or that cell. So most have, don't quote me on this pronunciation, but most have peptidoglycan cell walls and polysaccharide capsules. So some notes that I have here are that, once again, prokaryotic cells do not have membrane-bound DNA. They don't have a technical nucleus. So that's a huge difference between prokaryotes and eukaryotes. Bacteria are prokaryotes and do not have any membrane-bound organelles. Prokaryotes are generally smaller, and I said simple earlier. You could also say they are less complex than eukaryotes, and they do not have a nucleus. Lysosomes are the recycling center. Mitochondria capture and release energy, and the nucleus contains all of the genetic information. Ribosomes are the location for protein assembly. Plasma membrane separates the inside of the cell from the outside of the cell, so from whatever the cell is floating within. And we are going to hit more on all of these terms later within this chapter. I just wanted to kind of introduce them now as well. So another major difference between prokaryotic cells and eukaryotic cells is that prokaryotic cells are much smaller than eukaryotic cells. So on the left side here, you can see a prokaryotic cell versus a eukaryotic or like an animal cell. So that kind of just shows you the scale. Prokaryotic cells, from this angle, they look really simple in comparison to a eukaryotic cell. They don't have any of those membrane-bound organelles. They don't have a membrane-bound nucleus. They're just a lot more, technically, they're less complicated. They're more simple structured. So as a cell increases in size, its surface area to volume ratio decreases. So surface area actually limits life, and once the limit is reached, a cell will either divide or die. So that's a big part of this chapter, is that surface area does limit life. So this slide is labeled 2 cell types in domains. So let's look at the left here. I have a little image as well as just a statement. So bacteria are prokaryotes, and this is supposed to say with no nucleus. So like we said, they have no membrane-bound nucleus. There's nothing there holding that genetic material in a certain spot. While eukaryotes, on the other hand, the more complicated version and larger version, they are all all organisms are membrane-bound. They have organelles. And they have a nucleus. So this green image that's circled here is that nucleus. And what I mean by membrane bound is it's within that green area. So it's located within a specific spot in that cell, rather than just being free floating in any spot in the cell, like in the bacteria, the prokaryote on the left side. So now let's talk a little bit about eukaryotic cells, and we'll really dig into them because they are more complicated. So eukaryotic cells have membrane-bound DNA inside of a nucleus. So like we said, that's a huge difference. Really, it's the major difference between prokaryotes and eukaryotes is that membrane-bound DNA in a nucleus. So eukaryotes have membrane-bound organelles, which means that all of the organelles within the eukaryote are more organized and kept within a membrane. Plants, protists, fungi, and animals are all eukaryotes. So some of the notes that I have on this slide are that eukaryotic cells, once again, have membrane-bound DNA inside a nucleus. They have membrane-bound organelles. And once again, in this slide, we're kind of going to hit on some of the organisms or organelles that we want to talk about in the next few slides. So lysosomes within these cells are the recycling center. The mitochondria captures and releases energy. The nucleus contains genetic information. And the rough ER is actually a membrane studded with ribosomes. And rough ER stands for rough endoplasmic reticulum. So that's a membrane that's studded with ribosomes within the cell. So now let's really look at what some of these organelles look like. In the next few slides, we're really going to cover what they do. But I just want to kind of show you on this image what they would look like. They're all color-coded to kind of help with where they're located. So first off, this is the common eukaryote organelles. So this entire thing would be an eukaryotic cell. So something like an animal cell. So let's first off start with the nucleus, which would be right here. It's in red, and it's going to be this entire circle here in the center. So you have something called a nuclear pore along the outside of the nucleus. You have a nucleolus within the inside of the nucleus, the inner portion. Nucleoplasm within the central area of the nucleus under that nuclear envelope, which is the outer portion of the nucleus. So we can also talk about ribosomes. The word that people use is they stud things, so they kind of live on the outside of different organisms. Let's look at then the smooth endoplasmic reticulum, which is the area here that is orange. It also kind of stems this direction, but it turns into the rough endoplasmic reticulum right here. So that would be the green portion. They look really similar. The smooth portion is not studded with ribosomes, while the rough portion right here is studded with ribosomes. So if you kind of zoom in here and look at the outside, you're going to see little dots. That's those ribosomes studding the outside of the rough ER, which makes the surface rough, while the smooth ER is smooth on the outside. It doesn't have any ribosomes. So looking at the Golgi apparatus, it's here on the left side. Obviously, it won't always be located in the same spot. None of these things will, but they are membrane bound, so they at least stay within themselves and don't just free float in the cell. The last couple here would be lysosomes, which one of those is right here, a large portion of the cell, and then mitochondria, which are these blue guys. They're labeled here in pink just to show you where they're at, but you've probably heard of them back in high school biology. They are the powerhouse of the cell, right? So they're all about energy, and they're just kind of spread throughout the cell. So this chapter, once again, is about cell structure. So let's really start digging into each of these organelles within the cells that we've been talking about. And let's start off with, in my opinion, the most important, which would be the nucleus. The nucleus contains DNA, genetic material, which is the instructions for building proteins. This organelle is visible when looking through a light microscope. So in class, we're actually going to look at several different cells, whether that be plant cells, animal cells. We're going to look at them, and we're actually going to be able to see the nucleus under a regular light microscope. So in this image here, you have an animal cell. These are all of the nuclei, which is the plural word for nucleus. So they're a darker color, kept within a circle within these cells, and they're pretty easy to indicate in all of the images that we're going to look at together. So some of the notes that I have on this slide are that the nucleus is the primary storage site for DNA in the cell. DNA, we've talked about a lot in previous slides. DNA is composed of long strands stored in chromosomes. All cells contain genetic information, and chromosomes are long strands of DNA that are twisted and condensed. The nucleus is the largest organelle, which is why it's visible with a light microscope. It contains the cell's genetic material, which is the DNA. The mitochondria is a small compartment with an inner membrane where energy is stored and released, and many membrane-bound organelles are shown, including the Golgi that packages proteins into vesicles. So this kind of digs into other organelles that we're going to look at in slides after this one, but this slide is mainly about the nucleus, so just remember we're actually going to be able to see these in class together. So now let's talk about ribosomes. Ribosomes are actually the part of the cell where proteins are assembled, so they're extremely important. We've talked about in previous slideshows and slides that proteins need to be assembled correctly and folded correctly in order to work within our bodies. So how ribosomes work is that a messenger molecule will actually carry the instructions for building proteins to a ribosome, and within that ribosome, proteins are assembled. So, ribosomes read mRNA, which if you look at this image, this strand is going to be your mRNA. We've talked about previously DNA has a double strand and mRNA is just one. So this ribosome is going to read this strand of mRNA instructions, and then they will assemble amino acids in proteins right here. So they're using this amino acid tRNA, stamping it in on these what they call codons, and they're forming this amino acid chain or this protein using the instructions, which would be the mRNA strand. So whether free or bound, ribosomes are the location where proteins are assembled. They are very prominent throughout cells. They're found free floating. They're found stuck on different organelles. Basically, we want them to be easy to find within the cell because they're important to produce the proteins that we need for our body to function. Prokaryotes contain ribosomes floating in the cytoplasm along with genetic material, and then eukaryotes will have membrane-bound organelles, including a nucleus containing chromosomes, and they're generally larger. So ribosomes are extremely important units within the cell because they are where our proteins are assembled. So this is one of our kind of busy slides in this chapter. It's because on this particular slide, we actually hit on 4 different organelles that we really want to talk about. So let's talk about the membrane system of eukaryotes. So first in this slide, we kind of get into the smooth endoplasmic reticulum, or the smooth ER, which I have here in yellow. So like I said, it's located here. It's orange on the inside, tan on the outside, and the smooth ER does not have ribosomes studding the outside like the rough ER does. The smooth ER is smooth along the outside. It does not have any bound ribosomes, and it generates lipids, including phospholipids and steroid hormones. So then let's talk about the rough ER, which is in green right here. It looks very similar. The big difference is that it's studded with those ribosomes that we just talked about where our proteins are assembled. So the rough ER has a surface studded with ribosomes where proteins are assembled. The assembled proteins are folded, which is how they work, right? So you have to have a protein folded into the correct assembly in order to do its job. So within the rough ER, they're assembled proteins. They're folded into vesicles for transport. So a vesicle you can think of as like an Uber of the cell. So the vesicle picks up these folded proteins and then takes it to where it needs to go to work in our body. So most transport vesicles will deliver proteins to the Golgi apparatus, which is right here, and that's where further processing happens. That's not where all of these proteins go, but the vast majority go into the Golgi. So the Golgi apparatus is next. So the Golgi apparatus is sometimes referred to as the UPS of the cell, which is a really good metaphor because it receives packages. So it receives those vesicles often from the rough ER, the rough endoplasmic reticulum. And it receives the package, it modifies those proteins, and then it sends the materials both internally within the cell and externally out into our body where they need to work. So the Golgi is a very important organism, or organelle, I believe, to know. So lysosomes, up here in purple, they are small membrane sacs of digestive enzymes. The enzymes hydrolyze or break down macromolecules into smaller molecules for recycling. So if you're thinking of the Golgi apparatus as UPS, you can think of the lysosome as the recycling center. So anything that is considered, I guess, waste within a cell, the lysosome will recycle so that it can be properly used because the cells are very efficient. So the notes that I have on this slide are that all cells have an external membrane called a plasma membrane, which holds their contents inside. So if you're talking about the plasma membrane here, you're going to look at this big orange area on the outside. It holds their contents inside and determines which molecules are permitted to enter and leave the cell. So this membrane is considered selectively permeable, which means selectively it allows things to enter and leave the cell. It's not just anything in, anything out. It's very selective. The plasma membrane is composed of a phospholipid bilayer embedded with proteins, which we'll dig into later in a different slide. The membrane must take in enough nutrients to sustain the cell and must release enough to detoxify the cell. So it's a very delicate balance. It has to be able to bring in enough nutrients to survive, but it also has to detoxify. So it has to get rid of anything that it doesn't need because, like I said, the cell is very efficient and it doesn't want to hold on to anything it doesn't need. So the Golgi, like I said, is a vital system for packaging proteins. It's UPS. The lysosomes are small membrane sacs that are the recycling center, like I said. And mitochondria capture and release energy, which we're going to hit on in the next slide. And cytoplasm is a semi-fluid matrix that holds the organelles. So cytoplasm is sort of this orange area inside. It's that liquid that really holds the organelles where they need to go. And that's it for this slide. So now let's talk about the organelle that we all know and love and probably remember from high school biology, the mitochondria. So the mitochondria is the powerhouse of the cell, and that is my last point right here of this slide. It's the big thing that we all remember. The mitochondria are meant to capture and release energy. So they actually are responsible for making ATP, which stands for, and again, do not quote me, adenosine triphosphate, ATP. That's the energy within our cells. The cell's main energy carrying molecule. So the way I kind of like to explain that is think of it as money within the cell, right? So in order to do anything within the cell, ATP or money, I guess, would be needed in order to pay its way. So in order to make a transaction, to send something like a vesicle to the Golgi apparatus, to do anything important with those proteins like we talked about in the last slide, you have to have energy. So the mitochondria are the powerhouse. They create that energy. So if you're thinking of the Golgi as the UPS, the lysosomes as the recycling center, you can think of the mitochondria as the energy center of the cell, right? So you can think of them in that way. So now I want to talk a little bit about plant cells, because the eukaryotic cells we've really been talking about and that I've really been showing you are animal cells, those round cells with all of the organized organelles. Those are all animal cells. So plant cells typically are square. They have very rigid cell walls. So let's kind of talk about them. We're also going to see these in class while working with our microscopes in lab. So plant cells are also a eukaryotic cell. They have the common organelles that we just talked about. They also have additional organelles. So some of the things that they have that a eukaryote does not is they have an extremely large vacuole, which we'll talk about in the next slide. And they have chloroplasts, which we'll talk about in upcoming slides as well. So let's really talk about plant organelles. So some of the organelles that we've previously talked about, we talked about that membrane-bound nucleus, which right here would be that circle with the nucleolus inside with all that DNA. There's your nucleus. They also have the powerhouse. They have mitochondria. They have a large cell wall. Our cells, the animal cells, also have cell walls. They're just not as rigid in this square form. They have a cell membrane. They have cytoplasm. They have a lot of the same things that we have within our organelles. You can see here the endoplasmic reticulums. I believe these would be lysosomes. So they're very similar, but they do have two things that I want to hit on in this slide that we do not in our cells. So animals do not have these. So they have a large vacuole, which I think this image really does it justice. It's a very big, a vacuole could be considered like a vacuum. I kind of think of it. It's kind of an empty looking space within a cell. They are typically membrane-bound sacs that store molecules. Plant cells, though, actually typically have a large central vacuole for storing water for photosynthesis and helping maintain cell shape. So if you think about it, cells need what to live? They need sunlight, they need water, right? And so that's how they do photosynthesis. That's how they technically eat. That's how they live. So in order to hold water when there may not be a lot of water on the outside, they have a large vacuole within their cells that is meant to hold water for them. They also have chloroplasts, which I have over here in green. So chloroplasts have their own DNA, and they're found mostly in plants, and they contain chlorophyll. And chlorophyll is what makes plants green. So this organelle uses sunlight to produce food for the plants. So if you're thinking of just how plants are living and existing, that vacuole is their water source. This chloroplast is their food source. So lastly, I want to talk about the cytoskeleton. Now, I know previous slides we were talking about plant cells only. This is not unique to plant cells. The cytoskeleton can be found within all different cells. So a network of protein tracks and tubules functions include. So basically the cytoskeleton is made of a network of protein tracks and tubules. So those include the meaning for them would be structural support. They aid in cell division. So when a cell divides, they aid in that. Transporting organelles and cell movement as well. So the cytoskeleton has three major components. So they have, right here, they have microfilaments, they have intermediate filaments, and they have microtubules. So some of my notes on the cytoskeleton. The cytoskeleton is a network of protein tracks and microtubules. These proteins work to support cellular structure. They aid in cell division, transport, and moving cells. So cell walls are found in prokaryotes, fungi, some protists, and all plants. So like I said, this is not unique to just plants. This can be found in prokaryotes, eukaryotes, animals, plants. They're considered extracellular structures because they're on the outside of that cell membrane, and cell walls protect, support, and contain cellulose. So I was kind of talking about the difference between animal and plant cells. Plant cells have a couple more organelles that we don't have. They also have a very rigid cell wall. So they have a very rigid cytoskeleton outside of them that makes that shape that's almost a square. So all cells, whether prokaryote or eukaryote, are filled with a gel-like substance called cytosol. It's part of the cytoplasm. It's thick, like a soupy consistency of molecules containing many molecules necessary for cellular restrictions. The cytoskeleton is composed of three primary protein structures, which we already talked about, but those are the microfilaments, intermediate filaments, and microtubules, and they allow for movement. Once again, they aid in cell division and support the structure of the cell overall, which allows for cell transport. Okay, so we are to our key takeaways section. So I have 4 key takeaways for you at the end of this PowerPoint. So first, I want you to take away that light and electron microscopes allow us to visualize cells. Remember that light microscopes are what we will be using in class. Scanning electron microscopes, or just electron microscopes in general, they are very sophisticated pieces of equipment that are mostly used for things like medical research. Next, we'll go over here to prokaryotes do not have a nucleus, but eukaryotes do. So that's one of the main, big differences between our bacteria and archaea, our prokaryotes, and our eukaryote cells. So our plant and animal cells, fungi cells, is that prokaryotes don't have a nucleus. They don't have a membrane-bound nucleus, but eukaryotes do. So they have a very specific place where DNA is stored and genetic material. So then let's go here. All eukaryotes also have a variety of organelles. So ribosomes, endoplasmic reticulums, both smooth and rough. ERs, they have Golgi apparatus, which is the UPS. They have lysosomes, which is the recycling center. And then they have mitochondria, which is the energy house of the cell. And then in addition to these, plants also have vacuoles, which is that stored water source, and chloroplasts, which is where that photosynthesis takes place and where they get their food. So these are the 4 key takeaways I really want you to know. Chapter 4 is actually the last chapter in Module 1. So now you've covered chapters 1, 2, 3, and 4. So you're ready for your first exam. And there will be more information on that on Blackboard, but always feel free to send me an email if you have any questions. And thank you for listening.